This video will concentrate on cellular respiration, everyone's favorite topic in Biology 1440. This is a process that takes place in the cells of both eu and prokaryotes, but in eukaryotes it happens in an organelle called the mitochondrion. The mitochondrion is a very interesting organelle in that it appears to be derived from prokaryotes. We have a nucleoid in our mitochondria, which is a ring of DNA, just like in bacteria. We also have ribosomes inside our mitochondria that are similar to those in bacteria. We have an outer and inner membrane, like many kinds of bacteria anyway. Outer membrane, there we go. It's going to be a little crude, but it's me, so you should be used to that. And we have a matrix inside here. So let me label all of these things. So that's the matrix. This membrane here is the inner membrane, which I'll abbreviate as IM. Inner membrane, outer membrane, matrix, and the space between is called the intermembrane space. That's the structure of a mitochondrion. So remember, they appear to have originated from bacteria through the process of endosymbiosis, and the evidence for that is the nucleoid present that's similar to bacteria, the ribosomes that are more like bacteria's ribosomes than the ribosomes in the rest of your cell, and mitochondria split in half like bacteria do to divide. So mitochondria are one of those organelles that are not part of your endomembrane system. They have a separate origin. So the mitochondria is going to be responsible for a lot of redox reactions, the transfer of electrons from one molecule to another. And if a molecule loses its electrons, it often falls apart. You'll lose parts. A lot of times what we're going to lose is a hydrogen and electrons. So if you lose electrons and hydrogen, you're generally oxidized. And if you gain electrons, you become more complicated. And usually you gain a hydrogen as well, you're reduced. So we're transferring electrons as well as any energy associated with those electrons. So in this reaction, C6H12O6 plus 6 oxygens yield 6 CO2s and H2O. C6H12O6, that glucose, is oxidized into carbon dioxide. It comes apart, its electrons are stolen, and water is formed. So oxygen is reduced, electrons are added to form H2O. This process, if uncontrolled, can be really violent. So if you take oxygen and stick it to hydrogen, the electrons in hydrogen are drawn to that oxygen and they lose potential energy, and that energy is lost as light and heat. It's basically combustion. So fire is needed to make water, basically. If you did that in your own body, if oxygen and hydrogen, or I should say oxygen and the electrons of hydrogen instantly met in your body, you would explode. So we need to control that, and that's what cellular respiration does. So if this were an unregulated reaction, if water was instantly formed and the electrons from hydrogen instantly went to oxygen, we'd have an explosive release of heat and energy. But in the case of cellular respiration, we're going to run those electrons down an electron transport chain, and they will release energy slowly every step of the way down these electron carriers, and that energy will be harvested to generate ATP, and eventually we're still going to get water at the end of that reaction. Two ways to build ATP in this chapter. There's another way in the photosynthesis chapter, but in this way, the first way is substrate level phosphorylation, which will involve the use of an enzyme, which will remove a phosphate group from some other molecule and attach it to ADP, adenosine diphosphate, creating ATP. So that's why it's called a substrate level, because they're ADP and some other molecule here that contains a phosphate group are substrates in this reaction. The other way is called oxidative phosphorylation, because oxygen is required. That occurs in the electron transport chain, and this is where most of your ATP is generated. So this process requires three major steps: glycolysis, the oxidation of pyruvate in the citric acid cycle, as well as oxidative phosphorylation, which takes place in the electron transport chain. Steps 2 and 3 here occur in the mitochondria. Step 1 occurs in the cytosol of the cell, the liquid portion of the cytoplasm. So the major steps in glycolysis: in the cytoplasm of both eu and prokaryotes, we have glucose coming in. We burn 2 ATPs to add activation energy. So these are going to add activation energy to the reaction. We're going to produce 4 ATP, so that's a gain of 2, because we burn 2, we make 4, we get 2 out of it. We're going to make 2 NADHs. NADHs are electron shuttles. They carry electrons. So NAD plus gains electrons and becomes NADH. So a hydrogen comes along for the ride. We're also going to make 2 molecules of pyruvate. Let me go back. 2 molecules of pyruvate. Pyruvate is a 3-carbon molecule, whereas glucose is a 6-carbon molecule, so it's half of a glucose. So we make 2 of those, and they go into the mitochondria now. That is where they're going next. So your book shows it like this. Glucose comes in, we add ATP, we get 4 ATP out of it using substrate level phosphorylation. That's kind of important to know. And we're going to get 2 electron shuttles, NADHs, out of this, and 2 pyruvates. So at the bottom is a summary of what's going on here. 2 pyruvates are going to be produced, 2 ATPs, and 2 electron shuttles, as well as a few other things. Water is going to be formed, of course ATP, and some hydrogen ions. Now those pyruvates go into the mitochondrion, and we're going to have a series of enzymes all stuck together called a multi-enzyme complex that is going to now oxidize pyruvate. We're going to break pyruvate apart, at least partially. It's a 3-carbon molecule. We're going to turn it into a 2-carbon molecule and add a coenzyme to it, forming acetyl coenzyme A. So as we see here, pyruvate enters the cell, generally through a process of active transport through a transport protein. We're going to split off this carboxyl group right here. It's going to form carbon dioxide. This is the same CO2 that you exhale. It goes to your blood, to your lungs, and out of your mouth or your nose. This acetyl coenzyme A enters the well, I should say pyruvate enters the cell. It joins with coenzyme A. We're going to produce an NADH here, so another electron shuttle, and we have produced acetyl coenzyme A. This is the step known as the oxidation of pyruvate. By the way, these electron shuttles make their way to the electron transport chain. We'll see that later. So acetyl coenzyme A has been formed. The next step is the citric acid, also known as the Krebs cycle. So the Krebs cycle, or the citric acid cycle, is going to occur in the matrix of the mitochondria. There are 8 steps. We're not going to learn all 8 steps. We're going to focus on the generalities here. So up here, once again, we have the oxidation of pyruvate, right? Pyruvate, a 3-carbon molecule, comes in. 1 carbon is given off as a portion of CO2 here. Coenzyme A comes in and sticks to this molecule. We make 1 electron shuttle, as we can see, and a hydrogen ion. And we're going to produce acetyl coenzyme A, which goes into the matrix, into the citric acid cycle. You can see we're going to generate more CO2 here, so these 2 carbons are going to eventually be given off as carbon dioxide. We're going to make 3 NADHs, so these electron shuttles. We're going to make another ATP through substrate level phosphorylation, and we're going to produce a new type of electron shuttle called FADH2, which is a reduced form of FAD. Then the last product of this reaction is going to be part of the reactant in the next round, which is why the citric acid cycle is indeed a cycle. So seeing this in more detail, we can see acetyl coenzyme A comes in. The coenzyme group is broken off. The 2 carbons join with this molecule known as oxalacetate, which forms citrate. And each step here has its own enzyme that catalyzes each step of these reactions. So these are metabolic pathways here, or this is a big metabolic pathway here. So citrate is converted to isocitrate, etc., etc. We produce NADHs, these electron shuttles. We produce 1 ATP per pyruvate. We also produce an FADH2, which is another electron shuttle. And finally, we regenerate oxalacetate at the very end, which reacts to the next acetyl coenzyme A coming in to the reaction. So for each acetyl coenzyme A, we're going to produce 2 carbon dioxides at this step, 3 NADHs, 1 FADH2. And I say electron carrier, probably it would be better if I called this an electron shuttle, so we don't confuse it for the parts in the electron transport chain. We're going to make an ATP through substrate level phosphorylation, and the last thing we make is a regeneration of oxalacetate, which will join with the next acetyl coenzyme A coming into this next reaction. So now we go to the electron transport chain. This is happening in the membrane of that inner membrane of the mitochondria, or in the plasma membrane of prokaryotes. So NADH and FADH2 are going to drop off their electrons, and they're going to go down a series of electron carriers, releasing energy as they go, and that energy will be used to make ATP. At the very bottom of this, so if we think of it like a stair step of potential energy, those electrons go down each of these carriers each step of the way, and at the very bottom is an oxygen waiting for those electrons, and when they join oxygen, we're going to form H2O. We're going to get around 28 ATP out of this process. So as we see, NADH drops off its 2 electrons up here at the top of the electron transport chain. These are a bunch of different proteins and things embedded in the membrane of the inner membrane of the mitochondria. FADH2 drops off its electrons a little lower, so you get fewer ATP produced out of FADH2's electrons as they release less energy on their way down this pathway. So at the very end of this, we have half of an oxygen, so of an O2, so 1 O waiting here, and these electrons as well as hydrogen join this O, forming H2O at the very end. This is the oxygen that you breathe. This is why you breathe oxygen, is to power the electron transport chain. So the question would be, okay, so what? Electrons are tumbling down this stairwell, basically, this free energy stairwell. And notice the electrons don't even go to ATP. So how in the heck do we make ATP using electrons? Well, the electrons themselves do not go to ATP. It's the energy from electrons that we use to indirectly manufacture ATP. And I'll just point out before I move this slide that this Q right here is CoQ10, coenzyme Q10, that you may see for sale in the drug aisle of Walmart. So one thing I need to point out, remember diffusion. High concentrations of a molecule have higher potential energy than low concentrations. So the energy released from the electron transport chain is actually going to power proton pumps. And we're going to pump lots and lots of hydrogen ions into the intermembrane space. So we have a high concentration out here of hydrogen ions or protons, and a low concentration out here. So remember, a hydrogen ion is basically just a proton because its electron is gone and it has no neutron. Okay, the only way for these ions to get back across is through a transport protein, and they can diffuse back with their concentration gradient back into the matrix of the mitochondria. That is how we make ATP. So, having a look here. Electrons pass down the electron transport chain. Hydrogen ions are pumped out here. So we can see that we're pumping hydrogen ions at every step of the way, making a high concentration out here. Now, what happens next is a process called chemiosmosis, the osmosis of a chemical, basically, through this very special protein here, this enzyme called ATP synthase. Hydrogen ions flow through here very rapidly, and the kinetic energy of the hydrogen ions is converted into the potential energy of ATP. So the energy of the flowing hydrogen ions actually turns this protein. It's like a protein motor, and that catalyzes the reaction of ADP plus a phosphate group, forming ATP. So just like water flowing through a dam generating electricity, hydrogen flowing through ATP synthase generates ATP. This same process will occur in the chloroplasts of plants, and you'll see in that chapter that same process, hydrogen ions, same protein, ATP synthase. It's a very old protein that was evolved before animals and plants separated. In the case of animals, hydrogen ions are pumped into the intermembrane space and they flow back to the matrix. In the case of chloroplast, hydrogen ions are pumped into the thylakoid space and diffuse out into the stroma, but the result is the same. ATP will be generated. But remember, plants also have mitochondria, so they're doing both of these reactions. Okay. Excuse my phone ringing there. All right, so I just want to point out that it's not just glucose that can be used in this reaction, but proteins, carbohydrates, and fats. But carbohydrates and fats have a lot more energy in them. Fats, in fact, have twice the energy per gram as carbohydrates. And we can break down the glycerol portion of a fat even without much oxygen around. But to break down all of these high energy molecules here in the fatty acid tails, you have to have oxygen. Notice that this step right here can happen without oxygen, but everything from here down requires oxygen. So if you don't have oxygen around, none of this will happen. Okay? Without that, none of that will occur. Now we can break down proteins. They are something we often harness for our own building blocks, and we also can get some energy out of them. And one of the waste products of that can be seen here. We make ammonia, which is excreted in our urine. So overall, we have glycolysis, oxidation of pyruvate, citric acid cycle, and oxidative phosphorylation. Glycolysis makes 2 ATP through the process of substrate level phosphorylation. Citric acid cycle makes 2 ATP. By the way, I'm talking about per molecule of glucose, not per pyruvate here. And oxidative phosphorylation is going to give us a total of around 26 to 28 ATP. So all told, we get around 30 ATP out of each glucose. So don't get confused from the previous slides. We generally talk about pyruvates in each of those slides. But remember, you multiply pyruvate by 2, and that tells you how much you get per glucose. And all of these electron shuttles that we made, NADH and FADH2, they all travel through into the mitochondria to the electron transport chain to generate all that ATP. So we take electrons from food, and we use those electrons to pump hydrogen ions across a membrane, and when they flow back through, we use that to make ATP. And ATP is what you use to think, feel, move, and maintain homeostasis. One other thing I just wanted to mention real quick is that without the presence of oxygen around, our cells will undergo fermentation. Animal cells will undergo lactate fermentation. Some organisms like yeast will do ethanol fermentation. And in the case of fermentation, pyruvate is produced. However, pyruvate can't go on to the next step of the reaction because there's no oxygen around. NADH is produced, but the electrons have nowhere to go because there's no oxygen around. So the electrons are given back to pyruvate, which forms lactate in the case of animals. In the case of organisms like yeast, CO2 is first given off, so we end up with a 2-carbon molecule from pyruvate. Then electrons are given back to that molecule from NADH, and we end up forming ethanol, so alcohol, the same stuff that's in beer. But if there is oxygen around, then pyruvate goes into the mitochondria, and the regular process of cellular respiration will occur. One last thing, we can regulate the production of ATP using non-competitive inhibition. as we see on this slide, ATP will actually inhibit one of the first enzymes in glycolysis, phosphofructokinase. It will actually shut this process off, preventing the rest of respiration from occurring. So there are various steps here, including also the production of citrate will also inhibit this step. So we can actually shut this process off if we have enough ATP in the cell, and turn it back on when we get low on ATP. It's very important that we be able to regulate our reactions so we don't overproduce and waste resources. So that is the chapter on cellular respiration. Check this video out. Also check out the video on photosynthesis, and work on any worksheets you may have. Watch Khan Academy videos. Work this out. It's kind of complex, but it's really not that hard as soon as you start figuring out the patterns involved.